what was interesting was that not only did the mice look better with treatment, but the mice at the end of the psilocybin treatment looked better than they did at the start of the experiment.
Welcome back to Longevity Roadmap. This is Buck Joffrey, and today a very, very interesting topic. I kind of ran into this article while I was perusing through some longevity stuff, but it was an article by Dr. Louise Hecker and her colleagues over at Baylor, and effectively the idea is that psilocybin, you may recall from magic mushrooms, for example, may have a role in longevity. And actually, the data they presented in that paper was pretty strong. It's pretty potent in terms of longevity benefits for human cells as well as in mice. It's an interesting idea. If you've listened to the show for a while, we've had people talk about psilocybin in the context of PTSD, in the context of, depression, that kind of thing. Usually these types of things that are, psychiatric, but this is a completely different angle. This is suggesting that there might be something in the active compound, the active metabolite, which is psilocin or psilocin, however you want to say that may actually be geroprotective. Not really sure how. There's been some suggestion, as you'll hear from this discussion, about potentially the role of telomere lengthening, which, by the way, I believe you should already have a show on. I just recently interviewed the guy who discovered the telomerase gene. So interesting reference point there. Anyway, fascinating conversation for those of you who particularly have an interest in psilocybin and mushrooms and microdosing and all that kind of stuff. And we will have that conversation for you right after these messages.
Hey, longevity enthusiasts, it's time to take it to the next level. I've been fine-tuning my longevity regimen for years, and I look better and feel better than I did a decade ago. In fact, my blood work is even better than it was back then. And it's all because of my data-driven regimen. And it's inspired me to create a course and community just for you. It's called the Longevity Roadmap, and I urge you to check it out. If you're tired of your belly fat, tired of being tired, or just want to optimize yourself for the next 50 years, visit longevityroadmap.com. That's longevityroadmap.com.
Welcome back to the program. Today, my guest on Longevity Roadmap is Dr. Louise Hecker. Dr. Hecker is an associate professor of medicine at Baylor College of Medicine, a Georgia Alliance distinguished investigator, and a research scientist at Atlanta VA Healthcare System. She holds a BA in biology, MS in cell and developmental biology, and PhD in applied physics, all from the University of Michigan. And her lab made international headlines fairly recently here with a groundbreaking study showing that psilocybin, is the active metabolite of psilocybin, psilocin, can extend the lifespan of human fibroblasts and improve survival in aged mice. So I definitely wanted to speak with her. Welcome, Dr. Hecker. How are you?
Great. Thanks for having me on the show.
You bet. And so this is kind of a intriguing thing. I just, it kind of popped up in my feed, and I was like, wow, we've talked about psilocybin for a number of things, that's been in the news. But let's start with this. I mean, obviously best known as a psychedelic. What made you suspect that it might have some relevance for aging biology?
Yeah, I mean, it all really just started with a friend asking me questions about psilocybin that I didn't know how to answer. And I just decided to do some reading on PubMed and learn about psilocybin. And I was kind of instantly intrigued by, what we know about this drug, that had been in so many different clinical trials. It's been showing efficacy for so many different disease indications. And so, how is it doing that? How is it working for so many different diseases? And so that was kind of my first point that I found there's very few drugs that work for so many different disease indications.
What were some of those, either anecdotal or preclinical things that kind of piqued your interest?
I mean, so it's interesting because as a controlled substance, one of the criteria to be a controlled substance is this potential for addiction, but yet it's being used to treat addiction. Um, so I thought that was interesting. It's being used for all types of like, anxiety, PTSD, depression, even chronic pain, demoralization. So a lot of different disease indications. And then I found this one hypothesis that this medical hypothesis that someone had put out there saying, maybe, because there's this body of evidence that supports that shorter telomeres are associated with negative psychological states like anxiety and depression, and longer telomeres are associated with positive psychological states. And we know from clinical evidence that psilocybin is doing something to these psychological states that maybe it's having some kind of quantifiable impact on telomere length. And so as someone who studies aging and injury repair, I thought that I could actually test that. And that's kind of where it started.
Interesting. So you showed psilocybin extended the lifespan of human fibroblasts, right? So basically a type of human cell. So tell us about those findings. Like what specifically did you do, and what did you find?
Yeah. So we started with, cells have a natural lifespan, just like people, and they can only divide so many times. So so you can essentially watch the cellular lifespan over time, and you can, use an intervention like psilocybin and just see how that impacts cellular lifespan. So, and actually we use psilocin, which is the active, as you said, the active metabolite of psilocybin. Because that's what the cells see in the body. And yeah, I was actually surprised to find that, because when you think of serotonin targeting drugs, you think of the brain, but actually most cells in the body express those receptors. And so we thought, well, since we already know a lot about what's happening in the brain, and we know a lot about clinical, outcomes, let's look at other cell types that hadn't been tested. So I happen to be a fibroblast person, so we had fibroblasts handy in the lab, and so we used fibroblasts. And fibroblasts are also a well-validated model of replicative senescence. So we know we have kind of this, we already have a pretty good understanding of what that normal cellular lifespan looks like in fibroblasts and when they peak. And so that the first experiment was simple, just treating cells with psilocin or vehicle and watching their cellular lifespan or quantifying their cellular lifespan. And what we found is that in a dose dependent way, so you give more psilocin, you have even higher extension of cellular lifespan.
Interesting. Curious if you measured other markers with that. It's funny that we were talking a little bit about telomere lengthening, because my last, literally, like I do yesterday, I spoke to the guy who discovered telomerase, and one of the things he was pointing out is that from birth onward, we know we don't make telomerase anymore,
Oh,
and
wow.
that actually the lengthening of telomeres is sort of a separate phenomena just based on cell cycles. And, people were finding that activities such as, hyperbaric oxygen or whatever, that's lengthening telomeres, it's actually because of the elimination of short telomere cells and sort of creating an average. So the point I'm trying to make is his research and what he's trying to do now is figuring out novel ways to actually trigger telomerase, because we are one of the species that actually telomerase is not active in repairing these, telomere lengths. Whereas, in a number of other species, telomerase is there throughout life, and those species don't get cancer, for example. So I was just curious. I mean, I'm sure you didn't necessarily look at telomerase, but if you looked at any other any type of other markers in these cells that might have indicated what was going on.
As far as telomere length goes, we measured average telomere length in these cells. And so that's just kind of a standard way that we can look to see how this is impacting telomere length. But we look at so many other different markers, like for example, P16, which is cell cycle arrest marker. We see that that's typically increased with age as cells, tend to stop dividing. You see more cell increase in cell cycle arrest marker like P16. So that was decreased. We saw evidence of kind of markers suggesting that they have better DNA repair mechanisms in place. We saw increased SIR2 expression, which is kind of known to be kind of this master, anti-aging marker. We saw that there were decreased markers of NOX4, which is a master regulator of oxidant production. So the cells had less oxidant production, and at the same time, they had higher NRF2 levels, which is a master regulator of antioxidant responses. So you have less oxidants being produced, more antioxidant responses, and then the net effect of that is decreased oxidative stress.
How much longer did those cells live?
Yeah, so I think with like 10 micromolar, and we repeated this in 2 different cell types. We repeated this in lung fibroblasts and then also in adult dermal fibroblasts. And results were really similar in both cell types. So I think 10 micromolar was somewhere around 30% life extension. And when you go up to 100 micromolar, you get up to like 50 something percent cellular life extension.
Wow, that's incredible. So how about the mice? Because you also looked at aged mice. By the way, the mice apparently do create telomerase. So
Yeah, I stayed away because mice and telomeres and humans, and it's very different. So I didn't want to get into that with the mice. Mice have much longer telomeres than humans do. So it's a more challenging task to use mice as to make that comparison in mice. What we really want to do is to look at longevity, but we weren't able to do that study because at the time, there just wasn't any evidence that this was going to work. It was just based on our unpublished cellular data. So we had a really difficult time getting that type of study approved. But what the Animal Care would approve was a study to look at how it impacts median survival. And so basically our endpoint was survival. Does this improve survival? And so we gave a high dose once a month to the mice. And we started out with mice that were 19 months old, which is equivalent of about 60 to 65 human years. And because we wanted this to be a realistic kind of intervention type of experiment. And what we found is that median survival for the control group was about 29 months of age. So when 50% of those mice had died, we still had 80% survival in the psilocybin treated group.
wow. Other health span measures, mobility, cognition, resilience to stress, things like that.
Yeah, so some of the things that we could visually see, and at the time I didn't really know how to quantify, but the mice visually just looked better. They had, mice, you can see in the control group when they start. I mean, the aged mice, kind of just like aged people, they have, patch, their hair is starting to get bald spots. They're having some graying, whitening of the hair. So they had some of that to start as we started the experiment. And what was interesting was that not only did the mice look better with treatment, but the mice at the end of the psilocybin treatment looked better than they did at the start of the experiment. So, the bald spot they had grew back hair, and the white hair they had grew back in black. So they had these kind of really dramatic phenotypic changes that, again, the only way I knew how to document was just photographic evidence.
Yeah. So there's no like biological clock, epigenetic data, that kind of thing.
We actually, okay, so you're getting into good questions here. I want to point out too, is I know like if you're thinking about this from like, what would I test? There's so many things that we wanted to test, but the reality is that we are restricted by funding, by things that we have to do this research. And at the time, I had a really tough time getting funding for this project. So I had submitted federal grants, and they just got blown out of the water because there was no evidence out there at the time. Things that were getting, I was getting ripped on for were like, well, it's a Schedule 1 drug. Well, I know it's a Schedule 1 drug. So, I mean, things I have no control over. So this was not, I wasn't able to get a large amount of funding. What I was able to get was pilot funding from Emory to do this kind of high risk study. And so keeping that in mind, I did what I could with the pilot funding that I had. And so the end goal was, let's look at survival, right? Which is what we did. Now, that said, with the remaining pilot funding that we had left, we collected as much data as we could. And so we collected, bulk RNA-seq from every organ. We did proteomic profiling in the blood. So we haven't published all of this yet. We did epigenetic profiling. So we have a ton more data that we're still sifting through that we'll put out at some point. But we do have some answers. I can't give you all the
yeah, but we have to wait. We have to wait for the publishing.
Wait a little longer. We have to wait. We're still, again, trying to get, now that we have the paper out, trying to get some additional funding, because it takes teams of people to do that. I'm not a bioinformatics person, so I have to hire a whole team of people to take the data that I collected and help me to sort through that.
Yeah, which, it's interesting. So what do you think your next step would be? I mean, the thing that comes to mind to me is, gosh, this should be something that, interventions testing program should be looking at. I mean, they're right.
I think that we know that the elderly have worse outcomes when it comes to so many different diseases. COVID is one good example that I think everyone can relate to. We're all sort of exposed to COVID similarly, but we know from epidemiology data that the older age that you are, the worse outcomes you have from COVID. And so, as a scientist, what I've tried to do my whole life is try to understand what specifically is different about that age individual and try to target that one thing. But maybe we should take a step back from that and think about, because the fact of the matter is, it's not just COVID. There's so many disease indications where you have worse outcomes with aging. And so one idea, kind of the big picture idea from all of this work for me, and the Holy Grail, is what if you can slow the aging process in general? What if you just forget about individual diseases and you focus more on slowing that aging process? That may help a host of all different disease outcomes, to be better, to have better outcomes. So that to me is like the Holy Grail of this research is, can we use this as an intervention to improve age-associated disease outcomes?
Did you guys look at any inflammatory markers? Because one of the things, and I think about like sort of this, this very broad decrease in morbidity and this increase in lifespan and decrease in various health problems, it makes me think of inflammation, right? And as you know, a number of the drugs that are out there, including rapamycin, which has been very celebrated as potentially as a longevity, a geroprotective drug. A lot of the major effects are around inflammation.
Yeah, we well, I can tell you that because it's unpublished data, that we are looking at that, and there are really interesting impacts on inflammation. And so hopefully we'll have that out in the general public very soon.
Yeah. Got it. Anything unusual in terms of the findings of the types of, the types of mice that were getting better responses? For example, you see again in some of the intervention testing program drugs that they're often sex-specific responses, for example. Anything like that?
Yeah, so that's one thing we didn't get to test. Again, this is like part of the kind of limitations that you have when you're dealing with small amount of pilot funding, is you have to kind of like pick and choose the battles and the things that you really want to test. And so for that reason, because again, we had limited funding. We didn't have the money to test both sexes. We didn't want to add that as a confounding variable with this. So typically when you see an aging study, it's like starting out with like 100 mice, and aged mice are very, very expensive. So without that type of funding available to us, we had to start out with 30 mice per group. So we didn't want to add another confounding variable being sex-specific differences that could exist. So we don't know the answer to that question. Something that we do need to test for sure. We use female mice in this study, but we certainly need to validate these findings using male mice.
Yeah. Have you found anything? I guess small pilot studies, anything sort of correlative in humans and in the literature right now? I mean, I know you mentioned sort of some specific things like PTSD and all that, but as it relates specifically to any of the, longevity related issues?
Psilocybin's been around for so long. And actually one of the things that we've recently done, we're also about to publish this, is take a look at the funding and publishing map of psilocybin. Like what has ever been published in the US on psilocybin studies and who's funded it. And what we found is pretty surprising, which is that there haven't really been that many psilocybin published studies in the US. I mean, like if you think about it relative to something else like resveratrol, for example, 20,000 studies. There are only about 300 plus studies ever in the US on psilocybin. And that's kind of surprising if you think about how many clinical trials it's been in and how long we've known about psilocybin. And part of that, a reason for that is because of the regulatory challenges associated with working with a Schedule I drug. So it's not easy to work with a Schedule I drug. It was something that I didn't know anything about when I started doing this work. I just thought I would just walk in the lab the next day and order some psilocybin. I didn't realize, there is like a long process before you can even have it in your lab. I mean, I'm talking like maybe even a year. You have to obtain a DEA license. You have to have a lot of regulatory controls in place. So there are, it's not like, again, like you can turn around tomorrow, someone say, I'm interested in this and let me just start doing research on this tomorrow. It takes a lot of time and energy and effort to get through these regulatory challenges in order to even be able to start testing it. And so that's probably one of the reasons that contributes to kind of where we are with our understanding of psilocybin is these Schedule I regulatory challenges are significant and they're high. So that creates this kind of barrier for how easy it is to do these types of studies. So that's one of the reasons why I think we just don't know enough about psilocybin yet. And again, it's being that what it's known for is its hallucinogenic impacts. Most of everything we know, really 99% of what we know about it is what it does in the brain and what we know from clinical outcomes. But outside of that, very few studies have studied anything else really.
Yeah, interesting. So, again, big picture, I think what you're just kind of give your vision on where you're going to, go from here in terms of like, obviously you've talked about, looking at it as something that again is ultimately what you're kind of describing as a geroprotective drug, right? So when you think about scheduling that out in terms of, this is what I'm going to do next, this is what I'm going to do next, what does that look like?
I think,
enhance this?
part of what we're doing now is sifting through all this data, this massive amount of data that we have collected already, and trying to really understand the epigenetic changes that have happened in those same mice, what's happening at the organ-specific level. Separate from that, I think we really need to define the optimal treatment protocol. Some of our unpublished data, we're finding that like the frequency of dosing is really important, maybe as important or maybe even more important than the dose itself. And so we're finding that if you change the frequency of dosing, you have dramatically different impacts even. So understanding the dosing, the optimal dosing protocols for geroprotection is going to be really important. So like what happens if we started with aged mice, that were equivalent of a 60-year-old, but what happens if you start it at the equivalent of a 40-year-old? Are you going to have, that much more benefit, or does it not matter? what happens if you give, instead of, we gave 10 doses, what happens if you give 20 doses? Are you going to have, is that going to change efficacy? Again, frequency of dosing, sex-specific differences, how early do you start? Is there a point at which, you're beyond the point of impacts? Like if you start at the equivalent of, human of equivalent of 80 years old, is it too late to have an impact? So we need to really better understand the optimal treatment protocols to provide geroprotection. And then I think from there, that's where I think it will get really interesting in terms of looking at how can you use this as an intervention that could then improve age-associated disease outcomes, the things that normally happen with natural aging, or when you are injured, or you don't think of this as an injury, but COVID is a lung injury. When you have an injury in older age and you have this kind of geroprotection behind you, or you've been doing this for a certain amount of time, can you improve those outcomes? We don't know the answer to that, but I'm really hoping that psilocybin has the potential to do that.
One real quick follow-up question to the dosing. With regard to how much you gave the mice, for example, was that equivalent to what you would call a microdose for humans? Or are they getting high doses?
So we measure in animals, we measure so we did know something about dosing, and we actually spent a lot of time thinking about dosing when we did this study. So one is because of prior mouse studies, we know what psychedelic dosing in mice, generally looks like in terms of ranges. So we tried to we wanted to give a psychedelic dose just because I think there have been more studies with psychedelic dosing versus microdosing, at least up until this point. So we know a little bit more about the clinical benefits from high dose versus non-psychedelic dosing. So we tried to also extrapolate a bit from what's known about those doses in clinical trials and what's known about psychedelic doses in mice. The other considerations, which is why this becomes, people are asking me a lot about how does this translate to humans, and trying to make translations. And I warn people against doing those types of things at this point, because one, this is the very first study ever. This study needs to be validated, not just by our lab, but by outside labs. That needs to happen. But also, there's so many considerations that go into dosing. Like mice have their metabolism is like 4 times faster than humans, right? So if you think about it, like it's not apples to oranges when you're thinking about like metabolism. And so we actually gave a higher dose in mice to account for the fact that it's so rapidly metabolized in mice compared to humans. So the dose regimen was something that we determined from a number of different things, from taking, understanding what is a psychedelic dose in mice, what's a psychedelic dose in human patients, trying to come up with a dosing regimen that would be somewhat comparable, and then metabolism and things like that. So again, it's not ready for direct human translation. A lot more needs to be done in terms of that, and then another thing that goes with that is the long-term potential harms. We don't know anything about, most of the studies that are in clinical trials are like 1 or 2 high dose treatment protocols. I don't know of a study, there may be, but I don't know of one off the top of my head, where they've given like repeated dosing of psilocybin over a long period of time in human patients. That we know nothing about the safety of that, actually. So we have to think about, when we think about clinical translation, not only do we have to identify what's optimal for efficacy and geroprotection, but we also have to take a much closer look at potential harms. Does this have, when you think about, improving cellular lifespan and cellular dividing order, the first thing that, one might think of is, well, could this have a potential risk, higher risk for cancer? We don't know. There are some like correlative studies out there showing that suggest that psilocybin, people that have taken psilocybin have less cancer risk. But again, this was like rigorously tested. These are like correlative studies. And again, the reality is no one's really ever tested repetitive long-term dosing, especially in the context of aging and what the potential harms of that are. So we would need to really rigorously test that and define that, make sure this is a safe treatment.
Fascinating stuff. I want to thank you so much for being on the show, Dr. Hecker, and, good luck with this. And we'll certainly be keeping an eye out to see what you guys are coming up with.
Thanks so much. Thanks a lot.
Thanks for listening. A quick reminder that while I am, in fact, a surgeon, nothing I say should be construed as medical advice. Now, make sure to include your physician in any medical decisions you make. And also, if you're enjoying the show, please make sure to show your support with a like, share, or subscribe.